Hello students, welcome to EPG Pathshala. I am Dr. Switi Shrimali from Department of Zoology in Acharya Narendra Dev College, University of Delhi. In this lecture, I will be dealing with the module from Immunology. The title of the module is Models of Immunoglobulin Gene Structure, Multigene Organization of Immunoglobulin Genes, DNA Rearrangements and Mechanisms. After going through this module, I hope that you shall be able to appreciate the need for the vast repertoire of antigen specificity, understand the various models for immunoglobulin gene structure, describe the organization of immunoglobulin genes, understand the principles behind combinatorial diversity, appreciate the importance of somatic recombination, differentiate between somatic and meiotic recombination, describe the role of allelic exclusion in ensuring the uniqueness of antigen specificity, and understand the factors and processes involved in somatic recombination. So I would like to begin by saying that almost all the vertebrates have both innate as well as adaptive immune systems. Immunologists realized early on that one of the most important effector molecules for adaptive immunity is the antibody molecule. An antibody is an immunoglobulin protein that is secreted from the B cells and released into the circulatory system. In contrast, an immunoglobulin molecule present on the surface of B cells is called membrane immunoglobulin. Compared to an antibody molecule, the membrane immunoglobulin molecule has an extra hydrophobic transmembrane sequence. When the membrane immunoglobulin recognizes an antigen, its associated specialized membrane proteins, the Ig alpha, Ig beta heterodimer, help in signal transduction to the interior of the cell. This assembly of membrane immunoglobulin with the associated signal transduction proteins is known as the B cell receptor or BCR. So why do we need such a big repertoire of antigen specificities? The adaptive immune system protects the animals against a wide variety of pathogens. The characteristic features of adaptive immune system is that it is specific and has immunological memory against the antigens. The ability of the animal to launch an immune response against a specific antigen upon re-exposure is crucial for the survival of the animal. So let's discuss the models for immunoglobulin gene structure. In the present age of high throughput sequencing and structural analysis, we have a much clearer picture of how the genes are arranged at the immunoglobulin loci and how the sequence variations are brought about. However, in the early days, the absence of sequence data and structure information made it particularly difficult for immunologists to come to a consensus on the issue of immunoglobulin gene structure. Thus, many models were proposed to explain the structure of immunoglobulin genes and the origin of immunoglobulin diversity. Some of these are germline theory, somatic variation theory, and two-gene model. So the first theory that we will discuss now is the germline theory. The proponents of this theory advocated that all the variations required for recognizing the foreign antigens were already present within the genome of the organism. They postulated that a large portion of the genome is dedicated to code for the immunoglobulin genes, and this in turn gives rise to B cells with a vast repertoire of antigen specificities. We now know that this is no longer true. But the salient feature of this theory is that it did not postulate the presence of any kind of mutation or recombination to account for the tremendous amount of antigenic specificities. So let us now discuss the second theory, which is the somatic variation theory. This theory postulated that the vertebrate genome in the germline does not encode for all the immunoglobulin genes by itself. Rather, a relatively small number of immunoglobulin genes undergo mutation and or recombination to produce the staggering levels of diversity in antigenic specificities. In the absence of sequencing data in early years, this theory was hard to substantiate with evidence. However, recent advances in technology have now provided ample evidence in support of this theory. Another interesting development in the field was the two-gene model. The basic idea for this model came from initial sequencing experiments of the immunoglobulin proteins. These experiments revealed that the amino portions of the different immunoglobulin proteins tended to be variable, while their carboxy portions were quite similar. To account for these findings, Dreyer and Bennett in 1965 hypothesized that each chain of the immunoglobulin molecule is encoded by two separate genes, one for the variable region and one for the constant region. They further hypothesized that these two genes are joined at the DNA level to form a single immunoglobulin protein. However, this theory received a lot of criticism because it was against the one gene, one polypeptide hypothesis that was prevalent at the time, and critics found it hard to envisage the possibility that two genes could code for a single polypeptide. However, in 1976, Nobumichi Hozumi and Susumu Tonegawa provided irrefutable evidence for the two-gene model by showing that the V genes and C genes join together at the DNA level. Hozumi and Tonegawa used 125 iodine 125 labeled radioactive probes for hybridization. These probes were derived from either whole kappa chain mRNA or from just the 3' end of the kappa chain mRNA. BAM-H1 digested DNA from embryonic cells showed two distinct peaks with BAM-H1 digested DNA and the DNA from tumor cells showed only one peak when hybridized to the whole kappa chain. This indicated that in the germline, the variable and constant region genes are on separate BAM-H1 digested DNA fragments, while in the cells expressing kappa chains, these genes are joined together. Thus, the authors concluded that in the germline condition, the variable and constant region genes are located at some distance from each other, while during lymphocyte differentiation, the variable and constant region genes are joined together at the DNA level to form a continuous stretch. This experiment and many such early ones have had a tremendous impact on our understanding of the immunoglobulin gene structure. Our knowledge about the structure of immunoglobulin genes has increased greatly in the past few decades. We now know that each immunoglobulin protein is composed of two heavy and two light chains. The heavy chain of an immunoglobulin molecule is always encoded by immunoglobulin heavy or IgH heavy locus, while the light chain may be encoded by either immunoglobulin kappa or IgK or immunoglobulin lambda or IgL locus. Further, each heavy and light chain is comprised of a variable region and a constant region. The variable regions of heavy chain in combination with those of the light chain determine the antigen specificity of the immunoglobulin. The constant region of the heavy chain, on the other hand, is responsible for effector functions like complement activation or binding to Fc receptor on macrophages, which is a process which triggers phagocytosis of the antigen bound to antibody. The variable region of immunoglobulin heavy chain is encoded by three different genes: V, which is short for variable, D, which is short for diversity, and J, which is short for joining, while that of the light chains is composed of two genes, V and J. As shown in this table, the locations and sizes of the immunoglobulin loci are different from each other. The IgH locus is about 1250 kilobases in size, is located on the short arm of chromosome 14 and has 38 to 46 V genes, 23 D genes, 6 J genes, and 9 C genes. The IgK locus is about 1820 kilobases in size, is located on the long arm of chromosome 2 and has 34 to 38 V genes, 5 J genes, and 1 C gene. The IgL locus is about 1050 kilobases in size, is located on the short arm of chromosome 22 and has 29 to 33 V genes, 4 to 5 J genes, and 4 to 5 C genes. The number of genes depends on the haplotype of the individual and thus can vary between individuals of the same species. For example, the IgH locus can have anywhere between 38 to 46 The IgK locus can have anywhere between 34 to 38, and the IgL locus can have anywhere between 29 to 33 functional V genes in case of humans. Besides differing in their location and size, the immunoglobulin loci differ in their organization also. This figure displays the schematic representation of organization of human IgK, IgL, and IgH loci at the DNA level. At the IgK locus, the V, J, and C genes are organized in separate clusters. At the IgL locus, the V genes are organized in a separate cluster, but the J and C genes are located together in 7 combined clusters. At the IgH locus, all the V, D, J, and C genes are organized in separate clusters. The double slanting bars in the figure represent the fact that these genes may be hundreds and sometimes thousands of base pairs away from each other. So far, we have discussed that immune loci are composed of multiple genes. But these multiple genes are only 7 to 8% of the vertebrate genome, and therefore, by themselves, they are not enough to encode the vast repertoire of antigen specificity that is actually found in our lymphocytes. So, how is the immune system generating so many antigen specificities? In fact, there are many ways in which the immune system does this, and one of the most important of these ways is combinatorial diversity. To explain combinatorial diversity, I'll remind you that the antigen binding site of an immune receptor molecule is composed of V, D, and J genes. As seen from Tonegawa's experiment, these genes are located at great distance from each other in the germline DNA. But in lymphocytes, these genes are located right next to each other. Now, this coming together of V, D, and J genes can occur in many possible combinations. This phenomenon of recombination of the V, D, and J genes such that a large number of antigen specificities can be generated using a relatively fewer genes is known as combinatorial diversity. By calculating the maximum number of possible ways in which the V, D, and J genes can combine with each other, we can obtain a fairly good estimate of the possible number of antigen specificities that the immunoglobulin loci are capable of generating. In this table, I am showing you how to calculate the combinatorial diversity of human immunoglobulin loci. The antigen binding region of the IgH locus is comprised of V, D, and J genes. Therefore, in order to calculate the maximum number of antigen specificities that this locus can generate, we multiply the maximum number of these genes at the IgH locus. And 46 multiplied by 23 multiplied by 3 gives us 6,348. Similarly, the antigen binding site of light chains is composed of only V and J genes. Therefore, to calculate the maximum number of antigen specificities for the IgK locus, we multiply 38 by 5 and that gives us 190. In the same way, for IgL locus, we multiply 33 by 5 and that gives us 165. For calculating the maximum number of antigen specificities for all the immunoglobulin loci combined, we need to remember that only one light chain locus is found in an immunoglobulin molecule. Therefore, we need to add the maximum number of antigen specificities for light chain genes, that is 190 plus 165, and that gives us 355. Then we multiply the 355 by the maximum number of antigen specificities for IgH genes, that is 6,348. Thus, due to combinatorial diversity, just 154 immunoglobulin genes in humans have a potential to encode for a staggering 2.26 million different antigen specificities. Isn't that amazing? Now, I should point out here that calculating the actual size of the repertoire for antigen specificity is not so simple, because in reality, all the recombination events are not entirely random, and some types of combinations are generated more frequently than the others. Also, other mechanisms besides multiple immunoglobulin genes and their combinatorial diversity play important roles in determining the variety of antigen specificity. These mechanisms are p-nucleotide addition, exonuclease trimming, and n-nucleotide addition. We will discuss all these mechanisms also in a little while. Now that we have discussed combinatorial diversity, we need to understand that this bringing together of immune genes is caused by DNA rearrangement. This DNA rearrangement occurs in B cells and T cells, which are somatic cells, and therefore is known as somatic recombination. This is different from meiotic recombination, which also involves DNA rearrangement but occurs during gametogenesis only. Other points of differences between these two types of DNA rearrangements is that somatic recombination increases the diversity in antigen receptors within an individual, while meiotic recombination increases genetic variation between individuals in a population. Another difference is that in somatic recombination, the DNA rearrangement is intrachromosomal. That is, it occurs between different genes on the same chromosome. While meiotic recombination, DNA rearrangement is interchromosomal. That means it occurs between homologous chromosomes. Another point of difference between somatic and meiotic recombination is that somatic recombination involves RAG and TDT enzymes, while meiotic recombination does not. Let us now understand the major events involved in somatic recombination. As we have already discussed, the rearrangement of immunoglobulin loci is restricted to the lymphocytes of B cell lineage only. Further, even within the B cell lineage, the recombination event stage specific and immunoglobulin locus specific. Thus, heavy chain rearranges before light chains, meaning that IgH locus rearranges first, and it's shown on the left hand side of the figure. Also, recombination between D to J genes, which is known as DJ recombination, occurs before recombination between V to DJ genes, which is called VDJ recombination. Once the rearrangement at IgH locus is complete, recombination begins at the light chain loci, which is IgK or IgL in case of humans. Since both IgK and IgL do not contain the D gene, therefore they undergo V to J recombination only, and this is called VJ recombination. As you can see in this figure, all the lymphocytes are derived from the common lymphoid progenitor, or CLP in short. The differentiation of a CLP to a mature B cell is a tightly regulated process with many intermediate stages along the way. The first stage after CLP is the pre-pro B cell stage, which is followed by a pro B cell stage. The cells in pro B stage differentiate into cells in pre B stage and finally mature into a mature B cell. So, how does the stage specificity correspond with the immunoglobulin locus specificity? The answer is that at the pro B cell stage, D to J recombination begins at the IgH locus. In the event of a successful rearrangement at the first allele, a heavy chain polypeptide is formed. This heavy chain polypeptide combines with the surrogate light chain and is expressed on the surface of the large pre-B cell along with the immunoglobulin alpha and immunoglobulin beta heterodimer. The polypeptide assembly thus formed is known as a pre-B cell receptor or a pre-BCR. The pre-BCR leads to intracellular signaling, which ultimately leads to cell proliferation. The pre-BCR also inhibits rearrangement at the second allele, and this phenomenon is known as allelic exclusion. A successful DJ recombination event is followed by V to DJ recombination event. And after the rearrangement of the IgH locus, the light chain loci undergo V to J recombination in the pre-B cell stage. As in case of the IgH locus, if the rearrangement of one allele is not successful, the other allele is used. On the other hand, in case the first allele undergoes a productive rearrangement, allelic exclusion of the other allele ensues. The light chain then replaces the surrogate light chain of the pre-BCR. This new kind of receptor thus formed is known as the B cell receptor or BCR and is expressed on the surface of mature B cells. I wish to emphasize here that this allelic exclusion ensures that all the BCRs on the surface of a mature B cell possess the same antigen specificity. Having discussed the various major events that occur during somatic recombination, let us now discuss the many different proteins that are involved in the process of somatic recombination. The most important of these proteins are RAG1 and RAG2, TDT, HMG1 and HMG2, Ku70, Ku80, DNA PKCs, RT-MES, XRCC4, DNA ligase 4, and Cernunos. As is clear from this table, these proteins perform a variety of different functions. You will also note that some of these are specific for lymphoid lineages. For example, RAG1, RAG2, and TDT. The proteins recombination activating gene 1 and 2 called RAG1 and RAG2 are a very integral part of the recombination process. The expression of RAG genes occurs in a stage-specific manner. The RAG proteins bind to specific sites called recombination signal sequences or RSS on the V, D, and J genes and help in introducing breaks into the DNA. As shown in the figure, the RSS are DNA sequences consisting of a heptamer, a spacer, and a nonamer sequence. The heptamer sequence CA-CA-GTG is a conserved palindromic sequence, while the nonamer AC-AAAACC is a conserved AT-rich sequence. Sandwiched between the heptamer and nonamer is a relatively non-conserved spacer sequence, which is either 12 base pair or 23 base pair in length. Corresponding to the length of their spacer, the RSS are called 12 base pair RSS or 23 base pair RSS. The RSS are found exactly downstream of V genes, exactly upstream of J genes, and both exactly upstream and downstream of D genes. Since the RSS are the sites where the RAG1 and RAG2 proteins bind the DNA and cleave it, therefore, they ensure that the somatic recombination occurs at specific sites only. This site-specific nature of recombination is very important as recombination at non-specific sites can lead to cancers. Now that we have discussed the major steps involved in somatic recombination, let us now look at the molecular mechanisms underlying this fascinating process. The basic molecular mechanisms underlying all the kinds of somatic recombination are the same irrespective of whether it's D to J recombination, V to DJ recombination, or V to J recombination. It is also same for B lymphocytes as well as T lymphocytes. Now, the first step in somatic recombination is the binding of the RAG1 and RAG2 proteins to the RSS, which are adjacent to the immunoglobulin genes. A general rule regarding the RSS is that the 12 base pair RSS recombines only with the 23 base pair RSS and vice versa. This is known as the 12-23 rule. This rule ensures that at the IGH locus, D to J recombination always occurs before V to DJ recombination. After they bind the appropriate RSS, the RAG proteins introduce a single strand break in the DNA between coding sequence and its flanking RSS. This break is introduced at precisely the 5' border of each RSS. Now, as a result of the single-stranded DNA break, a 3' hydroxyl and 5' phosphate end are generated. Next, the RAG proteins catalyze a trans-esterification reaction wherein the 3' hydroxyl group at the end of the gene launches a nucleophilic attack on the phosphodiester bond on the opposite non-coding strand. This reaction generates two ends, a coding end having a closed hairpin loop and a blunt signal end. These ends are held together by RAG in a post-cleavage synaptic complex, or PSC in short. Note that overall, two coding ends and two signal ends are generated for each recombination event. The two signal ends generated in the process are joined by DNA ligase 4 to form a signal joint. Results in the formation of palindromic sequences and is therefore known as p-nucleotide addition. In addition to combinatorial diversity, the p-nucleotide addition also contributes to diversity in antigenic specificity. The introduction of nucleotide variations, which occurs by p-nucleotide addition, exonuclease cleavage, and N-nucleotide addition at the junction between two genes during somatic recombination, is known as junctional diversity or junctional flexibility. Remember, this is different from combinatorial diversity. In case of light chain V to J rearrangement, the coding sequences of V and J genes after p-nucleotide addition are joined by DNA ligase 4 and other proteins involved in non-homologous end joining, thus generating a coding joint. So what happens in case of a heavy chain? In heavy chains, besides p-nucleotide addition, the rearrangements involve two more mechanisms. The first one is exonuclease trimming and the second one is N-nucleotide addition. Exonuclease trimming involves cutting of nucleotides at the 3' end. N-nucleotide addition refers to non-templated addition of nucleotides to the 3' end of the coding sequences. This process is catalyzed by the enzyme known as terminal deoxynucleotidyl transferase, or TDT in short. After all these processes are through, the coding sequences of the heavy chain gene D and J for DJ recombination and V and D for VDJ recombination are joined together by DNA ligase 4 and other proteins, thus generating a coding joint. One thing to remember here is that p-nucleotide addition, N-nucleotide addition, and exonuclease trimming do not necessarily occur in groups of 3 and therefore can lead to an out-of-frame rearrangement. This can lead to cell death. However, these processes increase the variety in antigenic specificity and therefore are beneficial to the organism. Once the recombination for both heavy chain and light chain loci has been accomplished, the mature B cells start expressing the B cell receptors on their surface. All the BCRs on the surface of a mature B cell possess the same antigen specificity. So students, let us summarize what we have learned in this module. The first thing that we have learned is that the immunoglobulin molecules are one of the most important effector molecules of adaptive immune system. The next thing that we have learned is that an immunoglobulin can be either membrane bound or secreted. The secreted form of an immunoglobulin is known as an antibody. An immunoglobulin molecule is composed of 2 heavy and 2 light chains. The next thing that we have learned is that each immunoglobulin gene locus consists of multiple genes clustered at particular position on specific chromosomes. The next thing is that the variable regions of the heavy chain is encoded by 3 genes, the variable gene which is V, the diversity gene which is D, and the joining gene which is J. The variable region of the light chain is encoded by just 2 genes, the variable or V gene and the joining or J gene. The next thing that we need to remember is that DNA rearrangements occur in a cell type and stage specific manner. Another important thing to remember from this lesson is that RAG proteins bind to specific sites called recombination signal sequences, or RSS, on the V, D and J genes and help in introducing breaks into the DNA. RSS are DNA sequences consisting of conserved palindromic heptamer, a relatively non-conserved spacer and a conserved AT-rich monomer sequence. The diversity of the immune system receptors is brought about by 5 different mechanisms. First is the multiple immunoglobulin genes. The second is the combinatorial diversity of those multiple immunoglobulin genes. And the next 3 processes come under the umbrella term of junctional diversity. And these 3 terms then are N-nucleotide addition, P-nucleotide addition, and exonuclease trimming. So 5 factors: multiple immunoglobulin genes, their combinatorial diversity, N-nucleotide addition, P-nucleotide addition, and exonuclease trimming. Now, this process of generation of a vast multitude of antigenic specificity is an extremely costly endeavor for the organism. However, the benefits of this process far outweigh the cost. With this, I'd like to conclude the C lecture. Thank you so much.